online. And just a couple of housekeeping announcements. Next week, we will have a in-person visit. So that would be usual. If you have, to people on Zoom, if you have any questions or comments, please write me a quick chat, like say yes, and be ready to answer your question or comment at the end when prompted. And let's start.
Good morning. It is my privilege to introduce Dr. Bryan Roth, the Michael Hooker Distinguished Professor of Pharmacology at the University of North Carolina at Chapel Hill and the Director of the National Institute of Mental Health's Psychoactive Drug Screening Program. Dr. Roth is a leading expert in molecular neuropharmacology with a career spanning over 30 years, focused on G-protein coupled receptors, particularly serotonin receptors implicated in psychiatric disorders. His groundbreaking research has illuminated the molecular mechanisms of psychedelics such as LSD and psilocybin. In 2017, his lab published the first high-resolution structure of LSD bound to the serotonin receptor, revealing key insights into how these drugs affect brain signaling. Dr. Roth's current work centers on developing novel, acting psychiatric therapeutics inspired by psychedelics designed to avoid hallucinogenic effects. His team's effort include identifying non-hallucinogenic psychoplastogens, compounds that promote neuroplasticity and show promise for treating depression, anxiety, and other mental illnesses. Beyond drug discovery, Dr. Roth also invented DREAD, designer receptors exclusively activated by designer drugs, a revolutionary chemogenetic technology that enables precise remote control of specific neurons in vivo, now used globally in over 1,000 labs to advance neuroscience research. Please join me in welcoming Dr. Bryan Roth, whose work is in reshaping the future of psychiatric therapeutics.
Greetings. Thanks so much. Can you hear me? Yes? Can you hear me?
Yes.
Okay, good. Great. Before I start, these are my disclosures. I guess the one thing I like to highlight here is that, so there was this mention that chemogenetics is used in 1,000 labs. It's actually more than 1,500 different institutions across 53 different countries. So it's thousands and thousands of labs are using it now. And all the work that my lab has done is supported by the NIH and DARPA. And before I start, I just like to point out that sometimes when people give talks, I always get the sense that they're trying to sell the audience on some approach or some method. And the only thing that I'm trying to sell today are my ideas, and I give them away for free. Okay? And what I'm going to focus on today is basically drug discovery. And the key question now in drug discovery, I think, is what is a solved problem for drug discovery? What is a solvable problem? And what are fundamentally unsolvable problems? And those are the things that I'm going to try to highlight today. And the outline is, what I'm going to tell you today is that finding compounds that interact with G-protein coupled receptors, this is a solved problem. And also that structure guided design of ligands with particular properties, which we call signaling bias, I'm going to tell you this is also a solved problem. But the big problem that we have for drug discovery, for all areas of drug discovery, is that predicting the drug target, ultimately the drug target that you want to design a medication against, and the structure of the clinical candidate. So predicting that target de novo and designing the structure of the molecule de novo is currently unsolvable. And I think that's ultimately where we want to get to. And I'm gonna tell you how we're trying to get there, or is it, I would say. So I'm going to tell you that finding compounds that interact with GPCRs, I'm going to just tell you this is a solved problem, okay? And I'm just going to give a little historical background of this. So I've been involved in drug discovery for many, many years. This is a screen that we did in my lab when I was at Case Western, you can see in 2001. And basically what we did this very simple experiment. I was interested in the time, as I still am, in identifying compounds that are selective for various serotonin receptors as potential therapeutics. So what we did, by meaning my technician and I, was we had a library of FDA-approved compounds, and we simply screened them against various serotonin receptors. And this is, a typical result here. We use robotics to do the screening. These are all done in 384 well format. And we found this drug that is an approved medication. It's called tranylcypromine. And it appeared to be an agonist at the 5-HT2C serotonin receptor. And this was interesting to me from my background as a psychiatrist, because I knew it was a monoamine oxidase inhibitor. But the interesting thing about this particular compound is that it's the only monoamine oxidase inhibitor that causes weight loss. All other monoamine oxidase inhibitors cause weight gain. And there was evidence at the time that this receptor might be involved in the regulation of appetite, okay? So using funding from the NIH and the screening center that I ran, we identified this compound in 2001. A few years later, actually, Alan Kozakowski, who was there at the University of Illinois, Chicago, contacted me because we had been screening some of his compounds. And he asked me if we had any interesting leads. And I said, yeah, we have this interesting compound. It's a 5-HT2C agonist. I think, with some medicinal chemistry, we can optimize it. So this was one of the first compounds here, reported in 2009, ultimately led to compounds that look like this. And you can see where the pharmacophore was retained there. And for those of you who are interested in psychedelic drugs, this compound actually resembles, to some extent, the psychedelic compounds called bromo dragonfly or dragonfly. So we developed this compound, and all of the preclinical and IND enabling studies were funded by the NIH. And we developed a compound that is selective for the 5-HT2C receptor. Key here is it has no activity against key anti-targets, one of which is the 5-HT2B receptor, the other is the 5-HT2A receptor. And it turned out to be a G protein biased super agonist. And this is one of the compounds. It turns out that the compound that's currently in clinical trials, the structure of it has not yet been officially revealed. But this is a compound that's in the patent, basically. And you can see that it activates G protein signaling, which we measure through calcium flux extremely well, but has very little activity against arrestin. And we designed it in this way because we suspected that our compounds would have to be given daily. And of course, agonists cause desensitization, but compounds that do not engage the arrestin pathway do not cause desensitization. And so what we found was that in vitro and in vivo, the compounds are agonists and not subject to desensitization that is currently seen with other agonists. And there's the structure of bromo dragonfly. You can see kind of similar. Okay. So at this stage, of course, we had to get pharma involved. So we successfully handed this off to clinical development to Bright Minds Biosciences. And just for full disclosure, I have nothing to do with this company, but multiple patents were issued in 2021. The lead compound was identified, BMB 101, advanced to phase 1 trials for Dravet syndrome. Turns out that these compounds probably are really good for treating seizure disorders. The phase 1 was completed successfully. Phase 2 was announced last year, was commenced at the end of the last year, and the results are to be announced in December 2025. And based on this one drug, the company currently has a market capitalization of $400 million. So this is an interesting company. Basically, they have one drug. It's my drug and Alan Kozakowski's drug. And our prediction is that if the phase 2 results are positive, this will be a $2 billion asset. So UNC is going to be rich if this gets acquired. Okay, so that just shows you that yes, you can do drug discovery, even in a lab that's not in industry. But you can see that it took a long time. And this is sort of the standard approach for drug discovery. Basically do physical screening, and then it's a lot of medicinal chemistry. And we wondered if there was a way to do this that was more efficient. And the idea was maybe if we solved the structures of these receptors, that would be helpful for structure-guided drug discovery. So that's what we do. And then we employ structure-guided drug discovery. And the approach we use these days is ultra-large-scale docking, which I'll describe here shortly. This was invented by Brian Choiquette and John Irwin at UCSF. My lab did the initial validation of this approach. And if you compare this virtual screening approach to physical screening, the hit rate is, in some cases as high as 40%. Whereas with a high throughput screen, you're lucky if the hit rate is 1 in 1,000. So you get this dramatic improvement in the number of active molecules. We actually do both. And then the active compounds are then optimized by medicinal chemistry for therapeutic development. Okay, so this is the basic approach that we use, and as I say, this was invented by a colleague of mine. And the approach that we take now is we take large libraries of compounds that do not exist in the physical universe. That's key. But these compounds, in theory, could be synthesized by a medicinal chemist. Okay, so these are compounds that are invented by the computer, but theoretically could be made. Okay, so we have these large libraries. They now number in the billions of molecules, soon in trillions of molecules. And then using sort of molecular docking and I would say a molecular docking approach that was pioneered by the Choiquette lab. Each compound is then docked into the receptor in silico in multiple conformations. Okay, and this is actually one of the key innovations here of the approach. So each compound is docked in up to 1 million different orientations and conformations. And then each conformation is scored for fit. And then the high scoring molecules are ultimately enumerated, synthesized, and then tested physically. And so you can imagine if we're doing and I'll show you some screens here where we're interrogating more than 1 billion compounds, where each is oriented in 1 million different orientations. These are extremely large data sets. Okay. All right, so this was the sort of very first success of this approach. So this was a high resolution structure of a GPCR that was solved by my lab. And to date, I think this is still one of the highest resolution structures that have ever been solved. So this was the D4 receptor. And it was solved to sub 2 angstrom resolution by X-ray crystallography. And after we solved the structure, so there's the structure there. And all of these little squares here, these are water molecules. And you can see the compound is there sort of toward the upper part of the receptor. And there's this interesting water-mediated interaction with the side chain here, which was new. We wouldn't have seen that. The thing that was key, though, is that this receptor is a member of a family of receptors. And historically, it's been hard to design compounds that are selective for one member versus the other. But what we found here when we solved the structure was that this compound was oriented here in this little pocket here, this new pocket. You can see this phenyl ring here in this pocket. And this little pocket was selective for the D4 receptor. In neighboring receptors, basically this pocket was occluded. And so the idea was then we have these compounds, we can enumerate them in silico, and then dock them to the receptor and make predictions about which ones will hit. And these are, you're going to see various compounds docked in the receptor here. And then compounds that were active were optimized by analog by catalog. Okay. And ultimately, this led to compound 924. And this is a selective D4 agonist. So that was really the first demonstration of the power of basically solving a new structure and then interrogating it with new molecules. And over the years, we have done many of these projects in my lab and others. And some of these are highlighted here. I'm going to talk today about some of the work with the 5-HT2A receptor, many other receptors. But before I do, I want to focus on this family of receptors here. These are what are called MRGPRX receptors. And these are receptors that apparently have evolved to sense pain and itch. And my lab has done a lot of work on these receptors, solving structures. And a year or so ago, or 2 years ago, we were contacted by Christopher Norn, and Eden, who were then postdocs in the David Baker lab. And they'd seen our structure, and they wanted to do a collaboration to see if we could take this structure and design basically mini protein binders de novo that interact with this receptor. Now, the key thing about this receptor family is an orphan receptor family. And the structures that are predicted by AlphaFold are wrong. Okay, so we had real structures, and the person in my lab that's working on this is Brian Crum, very outstanding structural biologist. And at the end of the day, David Baker's lab, Chris Norn basically was able to predict compounds that look like, or peptides, small peptides that look like this. You see, these are helical peptides that are predicted to interact with the receptor here in this way. And I think key to their design here is they don't use the entire receptor structure for their design. They just use the binding surface here. Okay, so this was their prediction. And they took this receptor MRGPRX1, which we had recently solved the structure for. Again, the AlphaFold structure predictions were wrong. So that's the predicted structure of the peptide. And then we solved the structure. And so Brian Crum solved the structure. That's the structure. And that's the overlay. And the overlay was essentially identical. So you can see a very small error. And this is a different peptide. This is predicted peptide 2. And this is the solved structure. And then again, that's the overlay. And again, the overlay is nearly identical. Okay. So this has been done now for many GPCRs, and they have a company now, Sage. What is it? I forget what the name of it is. That is doing this on an industrial scale. And this can be done basically for every GPCR. The problem, I would say, with the binders is we're not able to specify whether they're agonists or antagonists, but we can easily get binders for basically any GPCR. So that's a solved problem. Now, the bigger problem here is structure. Once we get a structure and a compound that interacts with it, it's been impossible to design a compound that, for instance, activates the receptor or blocks the receptor, either as an agonist or an antagonist, or has particular signaling properties. What I'm going to tell you is that I think this is probably a solved problem as well. And I'm going to enter this a little indirectly. First, going back to AlphaFold. And so I think everybody here has heard of AlphaFold. And AlphaFold is basically a program that predicts structures of proteins. And when it first came out, I was interviewed along with other people about what the potential utility of AlphaFold might be for drug discovery. And what I said was that it's not useful, basically, because it's easy enough to get a structure. And if you're going to do drug discovery, why use a model when you can get a structure? But we felt that it would be useful ultimately to test this hypothesis prospectively. Okay. And so here's the idea that we had. The idea was we would solve a structure that AlphaFold did not have access to. Okay, so it would not be part of AlphaFold's training set. And then we would have AlphaFold predict the structure. Okay, and we would then interrogate both the real structure and the model, the predicted model, with this library of virtual molecules, which now numbered 1.6 billion. Okay, so again, these are molecules that do not exist in the physical universe. They've never been made before, but in theory, somebody could make them. Okay, so we're basically, interesting experiment, we're basically docking imaginary compounds in the case of AlphaFold 2 against an imaginary structure. Okay, and so how did this work? Well, both of them worked about the same. The hit rate was virtually identical for the structure-guided interrogation versus the AlphaFold. And what we are ultimately looking for is these agonists here, and the hit rate was identical for them. So this was the cryo-EM structure with the G protein biased agonist. And our very best compounds, which are here, you can see that they are, if we look at this compound here, it's nearly a full agonist for the 5-HT2A receptor, has no activity at the 2B or the 2C receptor, and is a G protein biased agonist. And we've shown that exemplar compounds have antidepressant activity in mice, and clinical candidates have been identified after an extensive medicinal chemistry campaign, and IND results are pending. So I'm not going to show you any structures of our lead molecules, but I want to focus on this molecule here. So there were a couple of interesting things about this this project. One of the big things was the structures of the compounds that we got do not recognize, any compound that interacts with this receptor. But this compound here was particularly interesting, this 7757. Now, one of the questions that we asked was, so the experimental structure yields these compounds, the AlphaFold model yields these compounds. Is there any overlap in the compounds that are identified? And there was absolutely no overlap. So these are absolutely orthogonal structures. And when we did a deep dive in this, and ultimately we were able to identify more than 400,000 novel chemotypes that potentially interact with the receptor. And there was very little overlap in the chemotypes. So one big advantage of doing this, of using an AlphaFold model, is if it works, you end up getting a different universe of small molecules that you can ultimately choose to optimize for structure-guided drug discovery. So it greatly expands the chemical space, which is key ultimately for success. Okay. So one of the questions that we've had is, what are the pharmacologic properties of these predicted active compounds? And so my lab has developed lots of technologies to interrogate GPCRs. And so we took advantage of these technologies to thoroughly understand what the nature of these novel molecules are, or is, that we're identifying that bind to these imaginary receptors. And one of the key assays we use is this arrestin recruitment assay. This was invented by Richard Axel's lab. And it's very, very simple assay. Basically, you have any GPCR here, and then you hook a vasopressin receptor tail on the receptor. And this is an arrestin binding site. And then there's a TEV, if we go back here, there's this TEV cleavage site. And when the receptor is activated, arrestin will bind to this arrestin binding site. And the arrestin has a TEV protease, so it cuts the TEV cleavage site, and then releases a transcription factor, which ultimately yields luciferase. So basically, this is a way of screening compounds against GPCRs, where ultimately we have a light-based readout. And as you can see over the years, my lab used this assay on a sort of receptor by receptor basis for great success. And we decided to basically develop this for all GPCRs in the genome. And we use this modular design strategy. I'm not going to go into this in any detail. But we were able to basically create a library of around 90% of the non-olfactory druggable GPCRs. They were all expressed on the cell surface at high levels, and we did intensive validation. And this is the resource. This is basically a phylogram of the GPCRome. Wherever there's a circle here, we have an assay. Initially, we didn't have assays for the adhesion or the frizzle and taste receptors. We do now. And this can be purchased from Adgene for a modest price. So basically, we have this library of GPCRs numbering 320. And we can now screen compounds against this library. And so now we can go back and look at the compounds that were identified from the virtual docking screen to see if these compounds are selective or if they're promiscuous. And these are the results of a heat map here. On the right-hand side, we have drugs that we know interact with the 5-HT2A receptor. And each line here is a different GPCR. Where it's red, you have actives. And these are the docking hits here. And you can see that the docking hits basically were, I would say, as promiscuous or as non-promiscuous as known psychedelic drugs, with some exceptions. So known psychedelic drugs have off-target effects at alpha-2 adrenergic receptors. Our docking hits did not. Some of them do hit dopamine receptors, but they appear to be fairly selective at serotonin receptors. Okay. So what this tells you then is that the compounds that you identify from the virtual screening platform have good drug-like properties. Okay, so they're going to be optimizable. So we're able to identify actives, and then the question is, can we identify compounds that have particular signaling properties downstream of the GPCR? And to answer this, we invented another technology. Again, this technology was initially invented by Michel Bouvier's lab. And we're going to focus here on G proteins. And since activation of the G protein is the very initial step in receptor transduction, what we wanted to do was to make sensors for various G proteins so we can interrogate signaling downstream of the receptor. Okay. And the approach that we took is fairly simple. This is the structure of the heterotrimeric G protein. And what we do is we put luciferase on the alpha subunit here. And then we put a green fluorescent protein on the beta subunit here. And if I just go to here, I'll just stop here just a second here. There we go. And so you can see the luciferase here is in close proximity to the green fluorescent protein. And so it's a very simple assay. We just add luciferin. Luciferase creates light. This activates GFP, and we measure the GFP fluorescence. Now, the amazing thing here, which was not appreciated when these were invented, was that the initial step in G protein interaction with the receptor is the heteromeric G protein undergoes this huge conformational shift like this, okay? There's this big change in the conformation of the G protein. And so this separates those. And so now we can measure this bioluminescence resonance energy transfer for individual G proteins and individual subunits. And we are ultimately able to create this for 15 G alpha subunits, 5 beta, 12 gamma, to ultimately yield 900 potential combinations. And we get very excellent estimations of compound potency and efficacy for individual G proteins. Okay, so now what we can do is we can go back and looking again at psychedelic drugs. Remember, we had identified these are known psychedelic drugs We identified various receptors they interact with. Now we're going to take each one of those receptors. So we know where the receptors they interact with are serotonin, dopamine, and adrenergic receptors. And then we're going to interrogate them against every single G protein that they interact with, okay? And you end up with these large heat maps here. And at the end of the day, what we're trying to make are drugs that target this one area of the signal transduction pathway here. These are 5-HT2A and GQ, okay? So known psychedelics are highly, highly promiscuous. We want compounds that are highly, highly selective. And we can identify these from ultra-large scale docking. Okay, so here's our compound. Here we got this extremely, extremely potent, highly selective, doesn't interact with any other receptors. And the question was, this was great, how the heck did this work? So what we did was we solved the structure. So we purify, have this compound, and then we were able to solve the structure of the 5-HT2A receptor with this compound, and that's the structure. And this is the predicted docking mode here, right here, versus the structure. And you can see that the big difference here in the two is that this tetrahydropyridine ring here is displaced a little bit in the docking as opposed to the structure. And the reason for this is it turns out that this tryptophan here is in a variable position in different structures. So it can basically flop up or down. And this causes enough of a clash here that this tetrahydropyridine ring basically goes to one place or the other. So it was ranked lower and that was ranked higher. So now, how can we use this information to identify compounds with signaling bias? Okay. So here's the approach. And the clue came from basically another GPCR. So Brian Crum in my lab had been studying the neurotensin receptor for many years. And we got this interesting compound, SBI 553 here. You can see here at the lower right-hand side. And this compound has this really bizarre pharmacology. So the neurotensin receptor can interact with multiple G proteins you see here. And this compound at GQ is what's called the negative allosteric modulator. And it's actually a positive allosteric modulator for other G proteins. And the key thing here about it is it is what's called a PAM agonist. So it has direct agonist activity for arrestin here. And it also is a positive allosteric modulator for neurotensin. So you have this bizarre compound, apparently is a PAM agonist for arrestin and a negative allosteric modulator for GQ, and then relatively silent for other G proteins. And we wondered, how the heck could this possibly be? And key, we actually want to make compounds here that are biased for one pathway versus the other. Okay, and this is important for some studies we're doing on making safer analgesic medications. Okay, so with Jeff Abe and Greg Shear, they had basically made analogs of SBI 553, not using any structural information. And they were able to find one here that you can see is a negative allosteric modulator against GQ, but is inactive against arrestin. Okay, so we have these really nice bias compounds. And the question is, how are these interesting compounds? How are they actually mediating their actions? And if we understand in some detail how they mediate their actions, maybe we can use this information for drug discovery. Okay. So again, what we had to do was solve the structure. So Brian Crum was able to solve a structure of the neurotensin receptor with GO in complex with SBI 553. And simultaneously, Eric Zhu's group was able to solve the receptor, same receptor, with arrestin with SBI 553. Okay. And so we compared these two structures. So here's the overlay here. Okay. So we're going to zoom in. And here's the key insight. Okay, so what I'm showing you here is this analog of SBI 553 that we made. Okay. And here is the parent compound, parent SBI 553, okay? And you can see that this analog, which is G protein biased, is tilted to the right, okay? See that? And this is because, very interesting, Chris will like this, because this residue interacts with a highly conserved residue in the G protein. This is in the alpha 5 helix. In this hydrophobic interaction between the receptor, the compound, and the G protein, stabilizes it for interacting with various transducers. Okay. Now, for the compound, when it's interacting with arrestin, it turns out that the finger loop of arrestin sort of sneaks in and directly interacts with SBI 553. See there? That's the overlay. Okay, there's the interaction. Now, the new compound basically stabilizes the GO interaction. And that's because we're now picking up some hydrophobic interactions here. You can see with this methionine and this leucine that are selective for GO. And that's going to be blocked basically with arrestin. And there were a couple of really, really important things here. The first is that, to our surprise, there were no large-scale differences in the orientations of any of the helices or any of the interaction surfaces. If we look at arrestins and various G proteins, that, you know, there are some differences, but they're not huge. But if we can target, basically, the key thing was targeting this compound directly for that receptor effector interface. Then we can begin to design compounds de novo that have particular signaling bias. Okay. So while this was ongoing, another postdoc, this amazing postdoc at my lab, Kugli Kim, or Yujong Kim, who's now on the job market, solved this structure with a taste receptor, TAS2R14. And to our shock and amazement, he found that this compound, it's sort of a standard bitter taste compound, this compound 28.1, also interacts directly at this interface between the receptor and the G protein, actually using the same leucine here, okay? And you can make, you know, we can make mutations which abrogate this effect. And based on this, then, so now in the taste receptor, we have this very similar pocket here, okay? And we wondered, can we now use this pocket for drug discovery? So we did ultra large scale docking against the pocket, okay? And we were able to identify compounds that are pretty potent, actually, and have actually solved the structure, of these new compounds in the pocket. And you can see a very nice overlay, very nice structures. And this is the story, basically. So we originally, when we originally solved the structure, we found that the tastant here is found here in this receptor G protein arrestin interface. And we actually found there was cholesterol here in the orthosteric site. So we think this is probably acts as a cholesterol sensor. So we have this interesting site, and then we are able to de novo basically design a compound that we predict would bind to that site. Okay. We tested it, and then it actually binds more or less as we predicted, and there are no changes in residue orientation. Okay, the binding pockets are similar. Okay, all right. Let's see how much time I have left. Okay, just a few minutes. Okay. So we're calling these compounds adhesives or molecular glues. And we wondered if one could do a parallel physical and computational screen to ultimately identify these. Now, the big problem in drug discovery, and I'm just going to end here so we have time for questions, is predicting the targets and the structure of the clinical candidate. De novo is an unsolvable problem. And to begin to answer this question, I invented chemogenetics. I'm gonna stop here because we're basically running out of time here, and just end here. So I just want to tell you how one of the approaches we're taking to identify. So as I told you, I think right now finding a drug for a GPCR is not a problem. It's a fairly straightforward thing. The big problem, though, is choosing the right molecular target for the right indication. And that's the problem. And the approach that we're taking, and this is from a freestanding research organization funded by the Eric Schmidt Foundation that my lab founded, called Eve Biosciences. And essentially what they're doing is they're screening all known bioactives and all known bioactive metabolites against all the druggable targets in the human genome. Eve stands for everything versus everything, so all drugs against all targets. And in theory, we think we're going to be able to identify all the therapeutic targets and their indications. And from that, ultimately, I think we'll be able to do de novo design and physical screening for precise control of signaling via these molecular adhesives. So that's one of the directions we're going today. I just want to thank all the people that were involved in this. They're all listed here. I just want to point out this fantastic postdoc who's now in the job market, Yujong. And then JK did the docking. Yorgo's lab was really helpful for us early on, getting cryo-EM going in our lab. And Jonathan, and of course, everything was funded by the NIH and by DARPA. And I'm happy to answer any questions you might have. Thank you.
Bryan, I will start with a few questions, really like always, fantastic and very stimulating. What a progress has been done in those few years.
Yeah.
We're talking about this adhesive and packet that you described at the end of your presentation. Wouldn't this be a way of selecting for arrestin versus G protein selectivity? And so then yeah, the problem of whether agonists or antagonists can be solved computationally and structurally, de novo prediction, whether that direction is viable.
Yeah. So let me just go back here just a second here. Yeah, so let's just go to the pocket here, this pocket. The goal is to use this information to design compounds de novo that either potentiate arrestin or G protein, or basically specific G proteins. That's the dream, but we're not there yet. Okay. So we're at the stage now where we can pretty well identify compounds that will, interact in that pocket. But we're not able to specify yet whether they will be selective for arrestin or for GQ or for GI or for GO. And that's, basically that's where we're going, but we're not there yet. But I think that's the final or an ultimate frontier here.
So my second question is, as this ligand selection and discovery progress so beautifully, there is remaining part of the drug development is the pharmacokinetics and also metabolism. It is kind of a high throughput way of determining that, for example, to prevent metabolism of those drugs so they can be a long-lasting or short-lasting, depending on the need.
Yeah, so that's the big, so as we say, the dirty little secret is we can make drugs. We can make, let me just say, we can make drug
ligands.
We can make ligands, right? Yeah, making drugs is a different thing. So what we do is the active compounds that are predicted from the docking are then computationally profiled. So there are Schrödinger, for instance, has this really nice suite of software that is, I would say it's not useless. It's useful for predicting metabolic liabilities, all that stuff. Okay, but ultimately, particularly when you get into novel chemical space, all bets are off, right? So this is the problem we have now. So I think we're ultimately going to get into the situation where we're going to be able to pick the right target at least, and we can get starting molecules. But then making those into drugs, that still is the big problem. Okay, and there are no shortcuts to that as far as I'm aware.
So let me ask a last question and pass to other people. All your screening, the library that you use, many of those compounds, you theoretically assume they can be made, but practically that's not correct, right? There are many compounds that you can draw the structure and you will never be able to synthesize, or they will be too difficult to synthesize, too expensive, or whatever other ways. So there's another approach here at USC where there's a modular building blocks that you can swap the quite large number of compounds, but those compounds are doable.
Yeah. So this is the synthon-based approach.
Yes.
And I'm a corresponding author on that paper. with
Then I will send you email.
Yeah, I'm on that. I'm a corresponding author on that paper. So the synthon-based approach works. I like this approach. Our success rate is 80% for if we, if we specify a compound that we want synthesized, there's an 80% chance that it'll be made by, we get them from enamine. So the success rate is 80%. So, the synthon-based approach works. It's not the approach I use. So.
Hey, Dr. Roth. My name is Vladimir Kefalov. I'm in the audience here in Irvine. Amazing talk. Thank you so much. I have two questions, if I may.
Yeah.
First, following up on Chris's question about this allosteric adhesion site. So if I remember, there is a high level of homology between the beta taste receptor and rhodopsin, and the G proteins also seem to crosstalk. So you can activate gasducin by rhodopsin, and also I think you can activate the G protein transducin by the beta taste receptor. So my question is, do you think that there is a similar adhesion site interacting with rhodopsin and its G protein transducin as well?
Yeah, we don't know. I've talked, so Arena Kufarina, I think, who's at UCSD, has done a big computational search for these pockets. And she thinks, as I do, they're probably in theory going to be everywhere. One of the things that we've done, we did recently, is there was a taste receptor that we solved the structure of. It didn't have a pocket. And so we did molecular dynamics simulations, and that revealed a transient pocket. And then we were able to use that for docking to find compounds that interact with the pocket. So what you might want to do is do sort of a, 1 or 2 microsecond MD simulation of the of rhodopsin with the G protein, and see if there's a transient pocket that forms.
Okay, thank you. And my second question is more general. You talked about the alpha, beta, and gamma subunits and all the combinations that they can form. So in photoreceptors, which are the cells that I study, there is a rod-specific alpha, beta, gamma G protein. But they seem to be fully interchangeable. So you can combine the rod alpha with the con beta and rod gamma, and the other way, basically you can make all combinations, and they seem to work pretty well. So my question is, what is your thought on the general role of the beta gamma in modulating or regulating the alpha signaling in different tissues and different cell types?
Oh. So beta gamma is really important actually for signaling. Let me just tell you 2 things, okay? So if you look at GI coupled receptors in the brain, the signal, so GI coupled receptors cause synaptic silencing generally in the brain. That's entirely due to beta gamma. Mm-hmm. There's absolutely no role for regulation of cyclic AMP in that. Okay, and it's through beta gamma regulation of snare formation and various ion channels. And it turns out for other G proteins, it's probably really important as well and has not been interrogated. So for GQ, for instance, we recently did, I don't have the data here, but we recently did quantitative phosphoproteomics. And what we found was that most of the, 2/3 of the signal was due to GQ, but about 1/3 of it was actually due to beta receptor kinase BARC, which is, of course, is translocated by beta gamma. So beta adrenergic receptor kinase, or G protein receptor kinase 2, actually is activated by beta gamma, and it turns out that it's phosphorylating lots and lots of proteins. So there is this role of beta gamma. I don't know in the visual system, because I don't study that, what the role of beta gamma is. But outside the brain, it's probably as important for signaling per se as the alpha subunit. It's just, it's been understudied.
Thank you. Just as a comment, I'm not aware of, and maybe Chris will correct me, but of role of beta gamma in photoreceptors. So it's assumed that alpha is the only signaling component, but who knows? Again, maybe beta is playing a different role that we still don't know. Thank you so much.
Yeah.
All right, let's move to Zoom. We have 2 questions.
So, Emiliana, can you ask your question?
I don't hear anything.
Yeah, we need
Emiliana, you may be muted.
Yeah, can you hear me now?
Yes.
Yeah, so thank you for this wonderful seminar. And my question is maybe naive, but how can you explain that the same ligand can be either an agonist or an antagonist at the same receptor? You know, some of these biased ligands.
So,
You know,
can,
depending on cell type, specificity, and region of the brains where they are expressed.
well, so I can explain everything. But what I like to tell people, so this is the sort of question I get a lot. And I have a blog here. If you send me an email, I'll send you the link to it where I go into this in a lot of detail. But I think the fundamental explanation for, you know, these weird things we see with ligands is if you understand this paper by Bob Lefkowitz, the extended ternary complex model, if you understand the math there, everything makes sense. Okay. I can't explain it any better than that. But if you really understand the extended ternary complex model, you can explain everything. So it's just, it all depends on relative receptor effector coupling and levels of both, basically.
But in looking for the specific ligands, like in those screenings that are made, how this is taken into account is not, of course, right?
Oh, we do. So what we're doing now is we are engineering. So we're well aware of this problem, okay? So we're engineering systems to take care of this problem. I'll just put it that way. We're engineering systems so that that's not going to be an issue. That's all I can say right now. Okay. It's ongoing.
Okay.
Yeah.
Thank you.
It's sort of complicated. I'm not going to get into it, but you know, this is always the problem, basically, and it's solvable, and I know how to solve it, and we're working on it. Okay. That's all.
Okay.
Okay. It would take too long to explain. And I have to go.
Yeah.
My wife is telling me I have my hair cut in 30 minutes, so I have to go.
Okay.
Bye.
Bye.
Yoshi.
Oh.
Bryan, we're not done. I'm sorry.
I have to go.
No, you're going to miss your haircut. You still have to ask me questions.
I have a quick question. So is it, oh,
Wow. Maybe it's kind of fun.
Now that is the way to build it. Not the mic and out. Wow. That was excellent.